Yes, this is a global workshop or webinar, and therefore I will say good morning, good day, and good evening to all of you, and welcome to the webinar, which is entitled Methods to Calculate Methane Emission from Stored Liquid Manure, and it's organized by the Manure Management Network, which is a part of the Global Research Alliance. My name is Sven Gede Sommer, and I'm a professor at Aarhus University, and I'm one of the co-coordinators of the network. At this webinar, three researchers with very much experience in developing models to calculate methane emission will give a presentation about their models and the methods to calculate the emission. So the first presenter will be Andrew van der Zaak. He is from the Science and Technology Branch, Agriculture and Agri-Food Canada, and he will present new improvements of the Tier 2 measuring method, calculation method of IPCC. Søren O. Petersen will come after Andrew, and he is from Aarhus University Department of Agroecology in Denmark, and he will discuss how mitigating methods for manure management can be investigated with the aim with the Arrhenius model and how it can be related to the model recommended by IPCC. And finally, Frederik Rastalby from Aarhus University, Department of Biological Chemical Engineering, Denmark, will present a new model for calculating methane emissions from liquid manure storages, which include microbial activity and growth. And then if you have questions, then please use the chat, and I will do my best to catch your questions and ask them to the presenters after they have given the presentations. So the first lecture is you, Andrew. The screen is yours.
Well, thank you very much, Sven. Appreciate the opportunity to be here and to talk about some of the work that we've been doing in Canada. I'll try to give an overview of measurements that we've done over the last more than 10 years and link that to the calculation approach in the IPCC 2019 refinement and highlight some areas where that could be improved potentially. So I'm going to start with talking about measurements. Something that I'm interested in is understanding the actual emissions that are occurring on farms, manure storages on farms. And Canada's an interesting place to do this type of work because we have a large country with a lot of different climates, but with some similarities in terms of the farm operations. So I want to highlight these three stars on the map. The one on the left is a dairy farm at about 53 degrees north in the province of Alberta. And that location has one of the shortest warm seasons, less than 150 degree days above 15 degrees Celsius there. We've done measurements there. This star on the right, that's one of the warmer parts of the country. It's where I'm located in Ottawa, Ontario, and we've done measurements there at a dairy farm. And so it has a longer warm season. And then the star in the middle, that's Manitoba, where we have a relatively large swine industry. And so that is where we've done measurements from swine operations. And so I'll talk about these three research locations and link that to the challenge of estimating emissions in simulations as well. But first of all, I'll start with the star on the map you can see in the top right. So this is in Ottawa, Ontario. This is a dairy farm with about 160 Holsteins that are being milked, and we've had this set up in the picture for over 10 years now. There's an open path laser measuring emissions. It's measuring the path integrated concentration of methane downwind of this manure tank. And I want to highlight that this tank is located in an area that has no other methane sources upwind. And so we also have a sonic anemometer that's measuring wind speed and turbulence parameters. And then we're taking that data along with the methane concentrations that are measured and doing simulations with inverse dispersion model to determine what the emission rates are from the tank. And that's being done every 15 minutes when the wind conditions are favorable. And we've done this for many years now. The annual temperature on average in this location is a little over 6 degrees Celsius, and this tank is typically emptied twice a year. And there's been a number of publications about this site. But the first thing I want to highlight that we learned or that we've observed from this farm and some of the temperature data that we've been measuring here is about how the temperature in the air increases in the spring more rapidly than the manure temperature. So there's a lag. Uh, and then the temperature in the air decreases more quickly in the fall compared to the manure. So we can see there's a significant shift in the timing of temperature between manure and air. And we've, with some colleagues, Tim Rennie and Ward Smith and others, worked on simulating this a number of years ago and to understand how manure temperature is impacted. And that's a fairly elaborate model, but I think the key takeaway here is that the difference, the manure temperature is impacted by a number of things, including some of the management practices. But definitely the manure temperature is shifted and lags behind air temperature. So we wanted to see that embedded in the calculations. And why that's important is illustrated here. If we look at this, this is 2 years of data from this farm. So the top panel is showing temperature in the air, which is the green, and in the manure, which is brown. And then the next panel below that, the second panel, is the total volume of manure in the storage, and then volatile solids below that. And at the bottom, methane production, which is peaking in August. And we can see that the peak air temperature, which occurs in July, is not when the peak manure methane emissions are occurring. The peak methane emissions are occurring in August, which is when we have peak peak temperature in the manure, as well as the peak volume of volatile solids in the tank. So we get a very sharp peak that year because we have an alignment between the temperature and the volume or the substrate. The next year, we don't have such a sharp peak. The peak is more gradual in terms of methane emissions, and that coincides with a misalignment. So we have peak manure temperature occurring in July, but peak manure volume occurring later in between September and October. And so when those two parameters don't align, then we get a more smooth peak in methane production. So that's just to kind of show the linkage between some of these key parameters that come into the model. And this one is to illustrate the relationship that we observed at this farm between manure temperature and methane production. And so the graph that is showing now is just highlighting the linkage of the linear relationship in 1 year, and how that at 15 degrees Celsius, the line intersects zero. So that suggests that there's a kind of a lower limit at which methane production is very minimal. And so with subsequent data, we have done some analysis with using degree days above 15 degrees as an indicator, like a heat unit. And so this graph shows the red lines are degree days above 15 degrees, and the black lines are methane emissions. And what we can see is generally a reasonable alignment between those two parameters at this farm. There's a number of things that have changed at the farm over the years, which affects why the fit differs, but I won't have time to get into that. But just to say, I think there's this is a useful indicator. Moving to a colder location in Alberta, this is another dairy farm, and here the annual mean temperature is less than 3 degrees Celsius. And here they're also removing manure generally once a year. Um, so what we can see here in this graph is pictures of the surface through the year, as well as the top panel showing temperature. The blue dots are the air temperature, which are increasing first, and the manure temperature takes a lot longer to increase. The manure temperature, which is the black dots, reaches a peak in September. And that, if we move down to the middle panel, which is the methane emissions, the peak methane emissions are coinciding with the peak manure temperature. And by the way, the peak manure temperature was a little bit over 15 degrees Celsius. So if we take those data and make an estimate for the whole year of methane emissions, we get a graph that looks like this. And then we've done that for 6 years at this same farm. And you can see an indicator by the photos of what the surface looks like in the summertime. And what stands out from these 6 years is that we had 2 years, 2015 and 2021, where there were much higher methane emissions than the other years. And those 2 years were when there were an unusual, extreme heat event that occurred there, a heat wave lasting about a week or up to 2 weeks. And those short-term extreme events seem to be linked to much higher methane emissions over the course of the whole year. And another thing is that those 2 years were also years that had very thick surface crusts. So a little bit more about the surface crusts is that we've observed at other farms too that when there is a presence of a thick crust like what's shown in the picture on the left, there's still can be very high concentrations of methane downwind. So this lends support to the idea that methane is finding its way through the crust, even if it is very thick. And I just wanted to say that I'll come back to the data I showed a moment ago on this slide later, talking about comparing the emissions with the IPCC calculation. Here I want to highlight the measurements that we've done in Manitoba at swine operations. This is unpublished data as of right now, but I wanted to highlight it because we have measurements from both the finishing barns, where we have the pictures shown on the left, and also from the outdoor storages, pictures shown on the right. And so we've done these measurements over a couple of years now. And so we can look at the annual emissions coming from the barns, where there's short-term slurry storage of up to a week. And then the long-term storage outside, which has a much longer retention time of about a year. But the temperature outdoors is very cold in the winter. It's frozen on the surface for many months, whereas the temperature indoors is generally warm through the winter, above 16 degrees. So what we can see in the bar graph, the blue sections of the bars are the emissions coming from under the barn, the short-term storage, and the red is the emissions from the outdoor storage, which are during the winter months and spring. It's similar or even more methane coming from the short-term storage under the barn, where it's warmer at that time. And in the summer, that's when most of the annual emissions occur, coming from the outdoor storage when the temperature warms up, gets above 15 degrees and sometimes above 20 degrees. So over the whole year, 84% of the emissions were coming from the outdoor storage, and that's really concentrated in a 2-month period. But it's still relevant to understand the contribution of both sources to the total. And I think as far as modeling goes, being able to integrate those two types of storage with different retention times and different temperatures into one simulation would be a valuable improvement. Another thing that we've observed from farms in this area were methane potentials. So the B naught parameter that was much lower than the IPCC default value. So we were getting numbers in around 335 or lower milliliters of methane per gram of volatile solids from the slurry coming out of the barn, whereas that's fresh slurry, not stored for a week. That's as fresh as we could get it. And the IPCC default value is 480. That's illustrated in the blue arrow on the graph on the right. So now briefly kind of summarizing how this relates to the and a bit of the background for the IPCC 2019 refinement. Table 10.17 is shown here, which here we have the MCF values shown for different climate zones as well as for different storage systems. But I want to highlight that this is all using the same calculation, all using the same calculator. The difference between these MCF values is due to the difference in retention time, so from 1 to 12 months. And the anaerobic lagoon definition is basically a long retention time. And then also the extent of removal is different between the liquid slurry and the anaerobic lagoon system, but it's all using the same calculator. So that was an important type of integration that avoids having to make judgment calls about definitions, which was the case in the 2006 version. Also, each of these climate zones is just using base, temperature data for monthly temperature data from each of them on average. And they're used to calculate graphs that look like the panel on the right. So we'll have a manure temperature, the volatile solids available, and the amount removed, and then the methane produced per month. And that cumulative methane from that is then compared to the methane potential to determine the methane conversion factor in the calculator. So the inputs to that calculator, there's two types. There's required input, which is highlighted in yellow, and then optional, which is more of a peach color. This monthly air temperature is ideally provided by the user, or even better if the user has manure temperature to just input that directly. But if air temperature is provided, then the manure temperature is estimated as lagging 1 month behind the air temperature with a minimum value that's set over on the right in the optional user inputs. The emptying efficiency is related to the manure removal timing, which is shown as a yes or a no here in column F. And the emptying efficiency, if it's known to be much less than 95%, for example, if only the liquid supernatant is removed from a storage, if the storage is not well mixed, then that emptying efficiency should be reduced, and that will have a big impact on the methane conversion factor. The idea here was to make something that was a little bit flexible, but also avoided some of those confusing definitions like MDP and anaerobic lagoon, which were in the 2006 version. Another thing I want to highlight is this VS entering liquid storage, which is in column I, and the second parameter underneath volatile solids excretion. That would be reduced if there's any kind of a separator or any sort of other treatment that removes volatile solids before it has the opportunity to reach the storage system. So again, one of the limitations here is that this is essentially assuming that all the manure that's produced goes directly into the storage, that there's only one storage. It can't really handle a treatment train with multiple stages, like an in-barn and an outdoor system. Here I'm just briefly comparing the observed values from the farm in Alberta, shown in the map on the right. This was the coldest location, and the the estimated values with the IPCC calculator. And that estimation has been done using manure temperature and air temperature. And generally for the 2015 year, the simulation is okay, but the observed values continue being high after the simulated values start to decrease. In 2016, which was a year without any, with a more normal climate, no heat waves, these estimated values were much higher than the observed. Again, remembering that the temperature here at this farm was rarely above 15 degrees Celsius in the manure. Overall, 6 years, the average simulation was an overestimate by the model. And if we look on the right, that's the cumulative values over the year. So when there was a heat wave in 2015, we get a reasonable agreement between the estimated value with the model and the observed. But in a more normal climate year, we're getting observations were about half of the model estimate. So I think this is about all that I had time for, but just to leave on these thoughts for improvement. So in colder regions, we've seen less methane produced than what the model is estimating. I think there's a methane sensitivity or sensitivity of the microbes to low manure temperatures, particularly when there's seasonal temperature changes, that the model is not calculating with the current Arrhenius function. And modeling manure transfer between storages, so a short-term storage under a barn and then a long-term storage outdoors, something that needs to be improved or added to a calculation approach. I think, yeah, some further potential improvements to the temperature function, particularly at low temperatures, and then the B naught values, which we observed were lower than the default values. And I'll leave it at that. Thank you.
Thank you very much, Andrew, for a very interesting presentation. I've not seen some questions in the chat until now, but I have some myself. So I would like to ask you about this B0 because the IPC says it's 480, and to me it's actually quite high. And you're also saying it is compared to yours. Is this something? Have you looked into if it's in general it's too high than if you have seen data from different parts of the world of barns or whatever?
I have seen some papers, I think from Denmark, that were indicating lower values related depending on the diet. So B naught's in the 350 range with certain diets, and so maybe that's the driver is
Mhm.
the improvement in the finishing diets. If you know in the last decades on commercial operations, there's a lot more precision feeding and other things that perhaps have changed fundamentally what the appropriate B naught value would be.
Yeah.
Um,
So that's, and then also about mitigation and reducing emissions. I could see you just proved that there's such a really big influence of temperature on the emissions. And you said finally, in the very cold climate, there's a problem with adaptations from cold to warm periods. So I
yeah,
wonder if you are thinking about if this could be kind of a mitigation option actually
Mm.
in many countries.
Like cooling, like a
Not
cooling.
cooling, but when you empty your storage and when you fill
Oh
it
yeah,
up and so on.
yeah. I think there's lots of it seems potential levers that could be used to reduce the annual emissions by having less uh, substrate in the storage during the warm times. But I know that the farm operations they seem to have many things on their mind, so many pressures. So it's maybe hard for them to prioritize this uh, compared to crop rotations and other things. Um, but yeah, I see the potential for mitigation of the outdoor emissions because they're so concentrated in a short period of time,
Yeah.
uh, to be a big opportunity.
At least it could be a cheap one.
Yeah, yeah, yeah. I hope so. And I think it's going to take some time to keep um, understanding better the processes and working together with the industries so that they can also have input into how it can be done in a way that is practical for each different
Yeah.
um, operational context.
Yeah. Thank you. And then there's a question from Joe Rudek, who's writing, I'm surprised by your results in methane emissions through crust. Do you have some comments to that?
Yeah. Yeah, so I didn't talk about it, but we have measured ammonia as well under those conditions, and we see that ammonia emissions uh, are very much reduced by surface crust. Um, and so I think it comes down to the properties of these molecules. So ammonia is very soluble, and ammonia and methane is not soluble. So if you have a soluble gas and you have a cover over top, then it will stay in solution. But if you have an insoluble gas and you put a cover over top that is not um, completely sealed, it will find its way out. If you put a sealed cover over top, then it will, like a biogas digester, it will inflate. It will increase the pressure and
Yeah.
inflate it. And so methane will basically find its way out is what I think. And
Yeah.
yeah, maybe there can be some oxidation occurring in the crust, like a biofilter. That's probably happening, but still the net effect on methane is we're not seeing a net reduction. I mean, we're not seeing that if you see a tank or a storage covered in a crust that it's going to have low methane. It might have very high methane. Maybe the bubbles for that's actually what's making the crust so thick. And maybe the
Yeah.
crust is preventing evaporation, and evaporation is a big uh way of one of the main ways of cooling the manure actually. So if you have less evaporation, you have a warmer temperature
Yeah.
inside.
Yes. Yeah. Thank you very much. I think there are no more questions. So thank you very much, Andrew. It's
All
really
right.
appreciated that you had time to come and give you this presentation.
Thanks,
S-
Sen.
So the next presentation is from Søren O. Petersen. So the screen is yours, Søren.
Thank you. Thank you, Sven, and share it here with you all. Okay, so I
This
hope you
is
can.
fine now. Yeah.
Yeah, great. Thanks. Yeah, so thanks for the invitation to give a presentation here. I think Andy's talk clearly illustrated the challenges of quantifying emissions. It takes a lot of hard work, and this was even with storages that were quite available for measurement. Often the storages are maybe closer to livestock buildings and so on, makes it more difficult. We need models. I think this is a fine opportunity to present and compare different ways of estimating methane emissions from manure. So in my presentation here, I'd like to briefly introduce methane emissions from manure management and talk about how the IPCC approach that Andy just introduced, how that compares with the alternative model that I would like to talk about today. Temperature response is also really important in our approach and also the response of methanogenesis to temperature. And in fact, I'd like to discuss in more detail what is the role of methanogens and how can we account for the methanogenic activity as it is influenced by local ambient conditions. And then I'll summarize. So if we look at methane emissions from agriculture and from livestock farms in general, the focus is on enteric emissions, always if you look to the literature. And that, of course, makes good sense at a global scale. Regionally, like in Europe or North America, with intensive livestock production systems, manure management is relatively more important. And if we then go to systems such as a pig farm, and you can see here that the methane emission from the manure is in fact the dominant source of methane and the target of mitigation. So we need to also always keep the context in mind and think about where is it relevant to consider methane emissions from manure management as the target of mitigation. The challenge of managing emissions of greenhouse gases from manure is that the variability is really high. There's a diversity in livestock production systems, and emissions are very variable depending on weather conditions, as we just saw. But if we want to manage for mitigation and also to document what we're doing, effects of what we're doing, it requires some robust estimates of emissions and effects of treatment or handling. And methane emissions, they are difficult to measure at farm sites with multiple sources, as I just mentioned, and so therefore we need a model. The basic equation behind the system that Andy just described is this equation. So basically, the calculation of annual methane emissions from a livestock category is based on the excretion of volatile solids during the year and the methane production potential. And then we have this MCF factor that varies depending on storage conditions and length of storage and a number of different factors. So these are the three basic components of the calculation. And this alternative model that I'd like to discuss today has the same elements in principle. We also have degradable volatile solids. We have a methane production potential parameter, and there is an Arrhenius type equation that accounts for the effects of storage conditions. And then how can the Arrhenius temperature response function explicitly represented? How can that help to characterize methane emissions from manure management? So I'd like to highlight these results from some 10 years ago, where we had a study done by a master student, Anne Olsen, who investigated the temperature response of different manure types. And you see in the Arrhenius plot here on the left that there was in fact very similar activation energy, similar slope for the different manure types, even though they were very different cattle slurry, pig slurry, and two diastates. So that would support the adoption of a common value for manure materials for activation energy. But then you can see the lines are parallel, which means that the intercept with the y-axis, which you can interpret as a methane production potential under ambient conditions, that could then represent maybe the methane production potential at the time of sampling. So it's not same as speed naught, which is kind of representing the freshly excreted manure. This represents the partly degraded manure that we sample. And recently I came across this review by Ralph Conrad on the temperature dependence of methanogenesis, and I thought it was really interesting. And here's a quote from the conclusion of the paper. He looked at activation energies for different processes, including hydrolysis, and he found that the values there are somewhat lower than the value that we determined here for manure, and even lower than methanogenesis in natural environments that have been reviewed in a large Nature paper. And these values are in fact quite close to the activation energy of 106 kilojoules per mole that you would normally see with pure cultures of methanogens. And his conclusion is that therefore methanogenesis in natural environments is normally not limited by hydrolysis, but rather by by the methane production step, that is the limiting rate limiting factor. And we have these main controls of methane production in manure. So the substrate, we have the storage temperature, and then we have the presence of the methanogens, because methanogens have to adapt to the manure environment. And this can be influenced by the chemical environment, the ammonia concentration, for example, or any additives that were given to the manure. And so the question that came to my mind is whether methanogenic activity, more than substrate availability, really is often the key control. Unless you have a very good inoculum that has been developing over the years, maybe you would be more reliant on the adaptation and the growth of methanogens in the environment. And I have a couple of examples I'd like to share. So one here is a new technology that is being investigated currently at Aarhus University by colleagues here. It's a washing robot, and you can see that this can be lowered in via a hole down below the floor in pig pens, and then you can flush the slats below the surface and also the sides of the pit. And you can see how 2 times of washing here very effectively reduced the total emissions of methane from the barn, and this was the emissions of methane from the manure that were reduced in this system with weekly export of the manure. So effective removal of manure and the methanogens in the manure appears to be able to suppress methane emissions inside the barns. But of course, we are exporting manure to the outside storage, so then we maybe just move the problem outside. So we also need some application of maybe some treatment of the manure in the outside storage. And we have been looking, as they have in Canada, we have been looking at slurry acidification for a number of years and how that influences methane emissions. And there are other methods also to reduce methane emissions from manure during storage, but acidification certainly is effective. You can see here how increasing volumes of additions of sulfuric acid from 1.2 to 6 kilograms per ton of manure, pig manure, effectively reduced methane emissions. So there's a need for combination technologies. We need to consider what happens in the barn as well as in the outside storage to have a really effective reduction. And then the question is, as also raised before, how can we quantify, how can we describe the contributions from the barn and the outside storage, which we need if we want to sort of simulate emissions and investigate mitigation strategies. And if we come back to the equation that I showed you before, we have the activation energy that we found in this previous study. We have the volatile solids, which we can quantify, or maybe we can look up literature values, but for the lignin, at least in the manure, which is not degraded under anaerobic conditions. And then we can calculate what is degradable volatile solids by VS measurements of the manure that we sampled. And the manure that we sampled from a source, we can take back to the lab and incubate, and we can determine a methane production rate at a defined temperature, which could be the temperature of the source where we collected the manure. And if we have this information, we can rearrange the equation and get an estimate of LNA, which is then the parameter that we think could represent methane emissions, the methane production potential of the sample that we collected. And with this approach, we can determine LNA for different point sources like pre-tanks or below slatted floors in the barn or in the outside storage with an incubation. And we did this in a European project with colleagues from Sweden and Germany and Netherlands a few years back. And then this paper came out of it that describes the method that I'm discussing here. So that was interesting that we were able to see differences between LNA values for cattle and pig slurry and for barn and the outside storage, and also some country specific changes. And with these 2 parameters, we can set up an empirical model with daily time steps, and in this way describe the emissions from barn and outside storage separately. We tried, of course, to compare with the IPCC methodology, and we have to say that we do get lower estimates using this approach that we did here, and we are quite sure about what the causes are. But we may have lower estimates with the model with the incubation method. In fact, we had an opportunity to compare with real emission measurements recently in a project on slurry acidification on farms. We collected manure samples at 1 meter depth in the slurry tank, brought them back for analysis in the lab, and colleagues investigated methane emissions from these covered storage tank using a tracer ratio method, where they inject the tracer and then look at the dilution or the escape of the tracer as well as methane, and then you can calculate methane emissions in that way. And in fact, when we compare the numbers here, we see we do get somewhat lower estimates from our incubations compared to the actual emissions that were measured on the farm. So let's just say for now that the Arrhenius model it probably underestimates methane emissions compared to on-farm observations. So we'll have to investigate this further. One thing I will say though is that when we have all these point measurements, it can also tell us something about what's going on inside the storage tank. And you can see here results from acidified and untreated slurry measured on. We had 8 different cattle farms and 8 different pig farms in this tanks in this study, and you can see that acidification quite effectively reduced methane emissions. But there were also some outliers, as you can see here, some pockets, well, first I thought it was maybe some analytical error in the lab when these outliers kept coming, and we could align them actually with the point in the tank where fresh manure is pumped in. Often this is where we would see the outlier. So now I'm thinking that probably some pockets of manure forms that is pumped into the tank that are not immediately mixed with the rest of the volume inside the tank. And so maybe for a period we will have an environment here that is very close to the fresh manure, and that's certainly useful information, and maybe this can help us to also the next step towards thinking about how can we avoid these pockets with fresh manure in a storage tank that is otherwise acidified. So there are, I think, still some benefits, some interesting things we can learn from taking point measurements and studying the spatial variability and also temporal variability at the specific points inside a large manure volume such as this. Yeah, and so to summarize, so the relevance of manure management for methane mitigation is region and system specific. Certainly we need to focus our attention also to the places where it really makes sense, and pig production, intensive pig production is certainly a system where we have a large potential for mitigation of methane via manure management. So the effective mitigation requires that we have methods to accurately estimate emissions and mitigation potentials, and a model, I think, with daily time steps is helpful in this respect. And we have defined this lab-based model that I presented, and it shares many features with the IPCC methodology, and we can calculate emissions estimates that could go into the inventory. But like I said, maybe it's currently what we are estimating is lower than what is measured actually. The methanogenic potential rather than the substrate availability could perhaps be a main constraint on methane emissions from manure. I think this is really significant and should be explored more, because the IPCC methodology assumes that this is the substrate and the temperature that are the main drivers of the emissions. And if we can somehow delay the growth of methanogens or inhibit them, then this may be maybe an effective way of reducing emissions. And this lab assay may not be able to represent methane emissions, but at the moment at least, and certainly it can help to characterize the sources and also to help quantify effects of management for mitigation. Okay, so finally, just acknowledgements. So at my age, there are so many people I'd like to thank here, so I will not give any names, but just say that many of the projects that we have conducted in recent years are part of policy support in Denmark. And so this is often funded by the Department here of Energy, of Agriculture, and of the Environment. And then also I'd like to thank all colleagues who took part in this M4 Models ERANET project a few years back during the Corona lockdown, but we managed to complete the project anyway. That was, I think, a nice collaboration. So thank you for your attention.
Thank you very much, Søren, for a fine presentation. And there was one in the webinar chat that disappeared again, but it was saying that it was a really great. It's now seen again to see that acidification has a huge effect on methane emissions. So that was a comment to your work. I'm not a biologist, so I would like to ask you now this ln to A, which you are saying is related to the microorganisms being adapted to the situation. Should one then change it over the year? Or you could see that Andrew was showing that if you have a kind of a temperature shock from winter to summer, it has a huge influence on methane emission. So is it, how would you recommend that this is used, the model in if when you go on with the work?
I think
I
that
don't know if it's a good question, but
no, I think we certainly need more measurements. We need to characterize specific management systems, and in fact, we have ongoing experiments now with pilot scale storage where we collected the manure on the farms, and we have big problems because we cannot really get methane emissions going. Often it's a bit erratic whether it starts or not, and if it starts, it's with a long delay. And I think it's because this manure came from farms where we took it from the pre-tank, and normally this manure would go to a biogas plant, so the retention time in the house is short. So we did not have adapted methanogens to start with in our pilot scale storage tank. So there are different ways. I think we need to explore further how we can delay or avoid the onset of adaptation and growth of methanogens.
yeah.
I can see there's also some questions here in the chat. There's one, acidification is one of the methods that can reduce methane emissions. What happens when acidified manure is used for fertilized farms? That's a good question. I also thought about that a lot. So I think there is a surplus of sulfate being added to the soil if we acidify according to what you would do normally to mitigate ammonia emissions. But in fact, that's why we are exploring now something we call low-dose acidification, where we add only 2 kilograms per ton of manure. And so that would fit the requirement from many crops. But in fact, what we note is that sulfate is in that case, the sulfate is in fact disappearing during storage. And that's probably because the pH reduction is not enough to inhibit sulfate reducing bacteria. So they are in fact active in the storage and will remove the slurry. So we don't have an excess of sulfate in that situation.
Yes.
And that's a question here about what was the temperature in the covered tanks when we compared the tracer ratio method with the lab model incubations. And yeah, this was autumn, late autumn, and even December, some of these measurements. So not ideal time of the year. And we tried to incubate also at very low temperature, even at 10 degrees or below, to match what happened, what was the conditions in the field. And that's also on the farm. That's also part of the reason maybe that we saw such poor activity in the lab. So again, we need to do more measurements also in the period July, August, where we see the highest methane emissions. And one last question here. In India, we do not have large industrial livestock farms where manure may be centrally collected. What aspects of livestock can help accurately determine manure emissions? Any studies to flag? I think I have to think about that, and maybe if you can send me an email, I'd like to get back to you on that. So okay, so maybe we should finish now, Sven. And
Yeah, thank you very much, Søren. I can't see these questions, so it's good you saw it.
In the chat.
Yeah, I'm looking at the chat actually, but it's well, thank you very much, Søren, and it's good you could see the questions, and thank you for a very interesting presentation that leads very well to the next presentation by Frederik Rask Dalby from Aarhus University. So, Frederik, the screen is yours.
Thank you, Sven. So I will just try to share my screen here. And
Okay, we can see it now.
so is it good now in presentation mode?
You could, yeah, but you could start it. Yeah, now it's disappeared
Oh, okay.
because now it's a join Zoom.
Do you see the window now with the?
Yeah, it's fine. Yes, I
Okay,
see it now.
let's go like this then.
Yes.
Right. So yeah, I would like to give a presentation of what we call the anaerobic biodegradation model, and that is also for calculating methane emission from stored manure. And it's a alternative that addresses some of the problems that Søren has talked about methanogenic activity. So I would like to start by showing some of the motivation for making this model. So you have already been presented with this equation, which describes the Arrhenius equation that is used in many inventories, and I believe that the IPCC also, what Andy he showed, is also some representation of this equation. And the thing about this equation is that it is chemically speaking, it's a first order reaction, which means that over time, as substrate is consumed, we will see a drop in emission because they are directly correlated. So you could imagine that you have a very simple scenario where you have manure stored in a tank, and you then keep the temperature constant, and you don't add any new manure. Then you would see a drop in methane emission over time as volatile solids is consumed. And this would not change if you had a very active inoculum to start with. It doesn't matter because it's not the amount of methanogens that controls the emission, it's the amount of substrate quantified as volatile solids. So this is not always what we see when we do these incubation tests in the lab, and not what we see in the field either. So we could imagine that a more complex model might do better. So if we use the knowledge that we already have about what's going on under anaerobic conditions, we can roughly separate it into 5 different steps here: the disintegration, hydrolysis, acidogenesis, and acetogenesis, and then finally the methanogenesis. And the 2 rate limiting steps are typically the first ones here, disintegration and hydrolysis, and then the methanogenesis in the end, because methanogens, they grow very slowly. The middle steps in this process, they happen rather quickly compared to the rest of the processes. So we might not need these in a model representation of this process. And therefore we can simplify this to a 2-step model, which only concerns the hydrolysis and disintegration, and then the methanogenesis. So what we came up with was the anaerobic biodegradation model, where you have some organic matter written here as particulate material, which will be degraded to volatile fatty acids using first order degradation kinetics. So that would be a similar process to the Arrhenius process. And this volatile fatty acids would then be further degraded to methane and CO2 by methanogens. So here we are explicitly modeling the growth of methanogens while they are taking up volatile fatty acids to produce methane, which means that over time we will accelerate this process of VFA consumption and produce more and more methane because the methanogen pool grows over time. These different processes, they are in this model, they are sensitive to inhibitors. This could, for instance, be sulfite or a lot of ammonia, nitrogen. But we also have regulators such as pH, and even the VFA itself can be inhibitive if the concentrations get very high and you have a relatively low pH. You could reach some inhibition processes. So as you see, this is a more complex model than what has been presented before, but it tries to reflect many of these processes that we know is going on in the manure. And now we are getting back to the first figure I showed, where we had a drop in emission over time because we had consumption. And here I'm trying to show that with the ABM model, you can actually represent two different scenarios. You can have the case where you have a decrease in emission over time because the system will be limited by the amount of substrate being hydrolyzed to VFA. That would be the green curve. It is kind of taking the same shape as the Arrhenius equation, which is the red curve. But you could also have, and you do have this in many systems, a situation where you don't have adapted or enough methanogens to produce very much methane. And in this case, you would see an acceleration of methane production over time with the growth of methanogens. So this would be a methanogenic limited situation. And what you would typically see is that you would reach some peak up here, and by this time, all the VFAs might be used up, and the system will switch to a hydrolysis limited system in this case. That is why it drops like this, the blue line. This is what we see pretty much when we incubate under controlled conditions in the lab. We have a increase in emission over time, and then we see a sudden drop by some point where the system change substrate. And this blue line actually represents, as Søren also mentioned, most of the natural systems that we have around where the temperature is not very high. Some of the situations we could think about here is, for instance, if you have fresh manure being excreted by the animal, yes, then you have some methanogens in that manure, but they are not really adapted to the colder environment they are excreted into, and therefore you would see that they are limiting the emission. You could imagine that you had a technology, for instance, like acidification, where you inhibit the methanogens, or you could also just in general imagine that the temperature is very low. In these cases, we know that since methanogenesis is very sensitive to temperature, that hydrolysis or that methanogens will typically be the limiting factor. The alternative case could be, for instance, if you have a biogas reactor, then you would have a hydrolysis limited system. And if you have adaptation over several years, then you might also reach a hydrolysis limited system. Yep. So in order to make this model work well with real experiments, we have conducted a lot of experiments in the laboratory where we measure and set up control conditions to try to isolate different parameters in this model. We typically look at responses to different temperatures, and we then have a continuous flow of air or nitrogen in the headspace of these flasks where we have incubated with manure. Then we have measured the rate of hydrolysis of different compounds and the sensitivity to temperature. We can also measure inhibition processes. We have measured what happens if you have feed spillage into the manure. This is something that we think actually happens to some degree in the Danish barns at least. And in this case, you would have a lot of starch that is easily degradable, which could spike methane emissions. And then we also measure other processes that consume carbon, for instance, respiration processes in the surface that produce CO2, but actually consume some of the carbon pool in the manure. Some of the things that we have seen in these experiments, for instance, in this experiment, we incubated manure over 280 days. at two different temperatures. So the blue lines here represent a low temperature, 10 degrees, and then we have red, which is 20 degrees. And the first point that you see, that is after the remaining mass of VS, in this case, after 280 days. And what's interesting here, I find, is that if you look at the lipids, we see that after 280 days, almost nothing is degraded of the lipids, except in one situation, that is when we have air in the surface. So it seems that lipids are mainly degraded when you have aerobic conditions in the surface and through that process. Then when we put the manure after to anaerobic digestion, we see that the lipids are quite easily consumed. So we see a large drop here. So this indicates at low temperatures, we don't see lipid degradation in the anaerobic phase. And then for the crude protein, we see that seems that a large fraction of the crude protein is actually easily degradable. You can see this from the triangle down here at 20 degrees. During storage, we have large degradation in the start, but then when you put it to the anaerobic digester, then you don't see much further degradation of the crude protein. So these experiments show that we have an easy and very hard degradable crude protein part in the manure typically. And we see this again and again in experiments we do in the lab. We also go out to full-scale farms. We have been in a lot of Danish pig houses where we measure emission, all the data sets up here and down to here. They are from pig houses with the finisher pigs. And we have then used the model to fit it to all these data sets at once. And we see quite a good performance for the pig houses. We also have some measurements for the outside storages, but these are more tricky to model, partly because we don't have that much measurement data. Typically, we only have maybe 6 measurements per year, and we have a very large uncertainty on the inputs that's going into the storages. So this is typically something, well, this is something that I would encourage when you do measurements on outside storages. It would be very nice to know what is going into the storage, not necessarily what is in it now, but what is going into it. Because this is the basis of all the process models actually. We also did some comparison to a Arrhenius model to see how do they compare. So using the same parameters for all these different full-scale measurements in general we get a better performance for the ABM model compared to the Arrhenius model, and we think this might be because we have the methanogenic component that can take over also. The downside of it is that you need to give this model much more inputs, and therefore it is also more reliant on the user of the model, what judgment they make. And we have made this web app where you can use the model. So this is our attempt to make some of these calls. So the user would have to say, well, what kind of flooring system are you operating with? What is the depth of the pit, and how much, when you empty the pit, how much manure is left? And then you can give a lot more information about productivity of the animals and what feed composition were given. And you can then upload this spreadsheet file to the web app, and you can process it and get the estimated emission. And you can also download the outputs. So this would be down here. You see an example of how the output would look from this web app. So I just want to dig into a situation where we have looked at 2 pig barns, and we are trying to look at this simple scenario where you have a standard way of removing the slurry, probably 2 to 3 times during a batch of finisher pigs. compared to where you remove it weekly. And here we see that in a pilot scale study, we predict that the reduction is around 50 to 60%. So we measure 53% reduction and model that it should be around 59%. But when we go out to the full scale and we do the same exercise, we see that the reduction that we measure and the reduction that we model is actually lower. And we think that this is because in the commercial farms, when you remove the manure weekly, you are maybe not doing it very efficiently. You are maybe leaving 10 centimeters of slurry left, and you could have also some parts of the pits where organic matter is actually accumulating in the sites. So this could contribute to the lower reduction effect in full-scale farms. And this is also a parameter in the model. So if you have a removal of manure, then you would typically see that the remaining manure would have more organic matter per volume, because particles they sediment to the bottom. And we have measured this at the farms, and you see typically between up to 150% enrichment in the organic matter in the leftover slurry, and actually new measurement that we have conducted shows even higher enrichment of the organic matter and the leftover. So this has a huge effect on the efficiency of weekly removal, which is now standard practice in Denmark, and we have not really. I don't think we have a very good confidence in the reduction factor that we're using so far, because it depends on all these things. So here I'm showing. The arrow here is wrong. I have shown some different scenarios that you can simulate with this web app. Out to the right here, you see a reference scenario, and you have the green zone, which is the enteric methane emission, and you have the blue, which is emission from the barn, and then you have the orange part, which is from the outdoor storage. And what you clearly see is that the outdoor storage is by far the main contributor to a methane emission, according to this model. And therefore, it is not enough to just reduce the storage time inside the barns. For instance, if you have weekly removal, which you see in the middle here, yes, then you reduce the emission by 50% in the barn, but you have in total on the whole chain, you only reduce by 12%, because you have a small increase in the storage. So you would need to combine it with some other technologies in the storage, for instance, biogas production. Then you could reduce in this example here, you can reduce the emission in total by 62% from the whole chain by combining weekly removal with biogas production. And then you can make that even more effective if you remove the manure weekly or daily, with scrapers. So there's still some work to do with this model, and one of the things that I need to underline here is that this is meant as a research tool where you can explore different mechanisms or management strategies on the farm to see what direction is the emission going in, and then on a more long term, we would like this to be maybe implemented in some real farm scale normative system values. Right now, there's some limitation in its usage because it depends on a lot of parameters, as you see in the Excel sheet that I showed before, and the user will need to make some calls here that might not be completely the way that we intended the modeler. So this needs to be improved from our part of. Then we need to find good ways of implementing mitigation technologies in this model. Some of them will be just straight reduction factors, but some of them will also be implemented directly in the process modeling. So for instance, inhibition of methanogens. And of course, we need to further verify the web app in order for this to get out and really have an impact. So that's what I had.
Thank you very much for this presentation. And I've been looking at your slides before, and now I understand them much better. So it was a very clear presentation to make me understand your work. Maybe I can come with. There have been some questions to Søren. One from Andrew. Maybe we should take them first, if that's okay. No, we'll take them after questions to you, Frederik, because that's so. Maybe I can just start. So when you have this, you have shown that there's an effect of cleaning the channels, slurry channels inside the barn. What about cleaning? And you say then you discharge your slurry into the outside storage in Denmark. What about cleaning this outside storage the same way? Or
Yes.
so you don't have adapted microorganisms. Could that be, instead of acidification or biogas production, an option?
Yeah, we are working on some technologies to do this. Some of the challenge has been to completely empty the outside storage. Usually there's like 40 centimeters of manure left, but we are working on some systems where we can reduce it to only a few cubic meters in a full scale tank, which is very little. We have not done any measurements on it yet to see if it works, and we are not completely aware about how large an effect the inoculum has outside where the temperature is normally lower, at least here in Denmark.
Mm-hmm.
So that would be interesting to see.
And then there's a question if this model, can it be used in, for example, in the Africa context?
Yes, but that it depends on what you mean by African context. Is that higher temperature, or is it in drier manure systems? Because the only thing that I could see that is that the temperature is probably higher in Africa. It is not fitted to systems that have relatively high dry matter. So it is mostly for liquid manure systems, and that might not be used a lot in Africa, but I'm not an expert on that.
Yeah. So you say it could be used for liquid manure in Africa.
Yeah.
If it's not too high in dry matter.
Yeah.
I think that's water flushing. May I ask when water flushing was applied, which model inputs or parameters were adjusted accordingly?
Yeah, so in ABM farm, you have the opportunity to apply water washing. So that would be, for instance, between batches of finisher pigs, you would typically wash the barn with water, and this would also flush out much of the residual manure, and therefore leave a less active methanogenic community for the next batch of pigs. So that's where this parameter comes into play.
And Andy was asking if additive or a cleaning product could have a similar effect to washing.
I would say yes. So the additive might inhibit the methanogens, whereas the flushing, that is more removal of methanogens and organic matter. So, but in terms of measurement of methane, it might have the same effect. Yeah.
And I think then there was I think we have to stop slowly. But if there are many questions for you, then I'm sure they can contact you. So there's saying, is this web app available for LMIC? I don't know what this abbreviation stands for.
I don't know either, but it's publicly available, so everybody should be able to access the link.
Yeah. So they can ask you if they can't find it.
Yes.
So yeah. I think there was to Søren a question about in India, the industrial livestock farm, but I think Søren was saying that they couldn't contact him directly, so it's not a question for you. So, but I wish to really, I wish to thank all three of you for some very good presentations, and I think they actually matched each other's very well. From Andrew's IPCC model that can be used worldwide and has to be simple, and Andrew was very open for improving it where it can be done. And Søren was focusing us on microbial activity, and you, Frederik, who have developed a little bit more complex model, but it's not very it's not like the big biogas model, but there are opportunities with it. I think it's at least the way I'm thinking. So I would like also, first of all, I would like to thank you once more, all three of you, for taking time to give this presentation. It has been very useful, and we have had quite a lot of participants during the presentations. And then I would also like to say that to the people who have attended the meeting, that they are welcome to send ideas for webinars to me, I'm the coordinator of this Manure Management Network, and we can talk about future webinars. But I would also like to bring your attention to the Emily 2026 conference. I hope all three of you are actually there, because I think maybe we have a session about the Manure Management Network where we will talk about how can we together how can we together work on improving the models and building and share data or whatever. We can discuss at that session. So I really hope all three of you will be there in Aarhus in September. It's Emily 2026. And of course, all other participants at this webinar, I welcome you of course, to come to the conference, which will be very interesting. Emily conferences have always been very good. And I hope you would then come to the session that is led by the Manure Management Network, and we could there focus on what we think the network could do to help people collaborate in the future. So thank you very much for the presentation, and thank you very much for listening, and then a farewell to everybody. Yeah. Oh,